Company Introduction. DAAN Gene Company Limited of Sun Yat-sen University was established in 1993 and is located in Guangzhou Science Park, Guangzhou, China. DAAN Gene was listed on the Shenzhen Stock Exchange on August 9, 2004, and has over 1,000 staffs, including about 200 doctors, graduates, and overseas scientists. DAAN Gene owned GMP Workshop, acquired the certificate of ISO 13485-2003 registration, C certificates, and CMD CAS for several varieties of diagnostic kits. DAAN Gene owned the self-owned intellectual property rights of the first fluorescent quantitative polymerase chain reaction diagnostic kit, and account for more than 60% market share of Molic microliterature diagnostic kits in China. Diagnostic kit for quantification of hepatitis B virus DNA. Principle. Cyber-Green 1 and Tachman probe are two common methods of fluorescence quantitative PCR. This kit adopts the Tachman probe method. Hydrolysis Tachman probe can be specific hybridization with the DNA template. The probe is labeled with the fluorescent reporter dye FAM at the 5-foot end and a fluorescence quenching dye TAMRA at the 3-foot end. When the probe is complete, if the probe is exposed to the light, the fluorescent signal emitted by the FAM is absorbed or suppressed by the quenching groups. They constitute the fluorescence resonance energy transfer. No fluorescent signal from reporter. During the PCR amplification, the probe is cleaved by the 5-foot to 3-foot exonucleus activity of the Taq polymerase, making the fluorescent reporter dye and the fluorescence quenching dye separate. Thus, fluorescence monitoring system can receive the fluorescent signal. A strand of DNA was amplified. There will be a fluorescent Molic microliterature formed. The fluorescent signal accumulation and PCR products formed in sync was realized. Two, preparation before experiment. Add 10 milliliter of absolute alcohol into the inhibitor removal buffer. Put a tick on the cap and preserve it at room temperature. Add 44 milliliter of absolute alcohol into the desalting solution. Put a tick on the cap and preserve it at room temperature. Add 1.375 milliliter of elution buffer into proteinase K. Mix them thoroughly and store the mixture at -20 degrees Celsius after dissolution. Carrier RNA centrifuge at 8,000 RPM for several seconds. Add 110 microliter of HBV internal control solution.